Welcome everybody and welcome to another PHA4GE webinar. For those that don't know PHA4GE, we are the Public Health Alliance for Genomic Epidemiology. So we are a collaboration and aspire to be a community of practice for the use of pathogen bioinformatics and genomic epidemiology in public health. And today we have two speakers. Patrick Ayscue from the CZ Biohub, where he leads biosecurity efforts and the COVID tracker project. He has a background working as an epidemic intelligence service officer at the US CDC, and where he was actually deployed to Liberia during the West African Ebola outbreak. And he has a background also previously at Metadata Incorporated, leading a team of international scientists building capacity and designing research studies in biosurveillance, biosafety, and biosecurity. He actually holds a DVM, Doctorate of Veterinary Medicine, if I understand correctly, from Cornell University, and where he completed a postdoc in quantitative epidemiology. Yeah, it was quite a long list of achievements. Really glad to have him here with us. And then Dr. Alli is a genomic epidemiologist at the Zuckerberg initiative, which shares some initials with the CZ Biohub, but is a different thing. She works on capacity building to help lab workers and epidemiologists learn how to use genomic data to explore infectious disease transmission dynamics. And additionally, she helps guide development of software tools that make genomic epi more approachable to public health practitioners. So welcome to both of you. And I will now stop talking so that you can start. Thank you.
Great, thanks, Peter. And thanks to everyone for joining and for having us here today. Happy to give you all a bit of a view into some of the work that we've been doing across the last year or so. Sorry, is that working? Are you able to see my screen there?
Yeah, we can see it, Patrick. Just have to go into present mode, I think.
Oh, there it is. All right. I was hiding the presentation from me, but now I found it. Okay, there we go. Awesome. Thanks, Alli. So I'll give everyone a little bit of context on some of the work we've been doing at Chan Zuckerberg Biohub and the Chan Zuckerberg Initiative across the last year and a half or so. Just to ground you a little bit in who we are and what we do and why we're engaging in this work and talking to you all here at PHA4GE. A little bit about the organizations. Chan Zuckerberg Initiative is Mark Zuckerberg and Priscilla Chan's primary philanthropic endeavor, and we have a science and infectious disease division there. That's where Alli contributes her work. And there we have a goal of determining, identifying the origin and spread of all infections by rapidly and effectively identifying and tracking pathogens at scale. A lot of this work historically has been in the field of metagenomic sequencing and disseminating those capabilities, both through capacity building and technology development. And we've been interested in applying that within the context of genomic epidemiology in the pandemic and beyond as well. Excuse me. I'm at the Chan Zuckerberg Biohub, which is also supported by Mark and Priscilla's foundation. We're a nonprofit research organization, and we have a rapid response team led by Christina Tato there, who many of you know. That's really focused on delivering capabilities to the front lines where they're needed for outbreak response. And that's how we came to a lot of this work as well. I joined the Biohub in February of 2020 and promptly worked on nothing that I expected to be working on and have been working pretty much exclusively on the COVID response since arriving there. Some of those early conversations when I arrived to the organization and we realized where the pandemic was heading were, how can we contribute? Where can we be supportive? Where are we uniquely positioned with our expertise to be contributing in this space? The Biohub has deep expertise in whole genome and metagenomic sequencing, technologies associated with that. Early in the pandemic, there was a need for high throughput testing, and we serviced that through the fall of 2020. And then in the summer of 2020, we also stood up what I've been characterizing as whole genome sequencing as a service for health departments throughout California. And I'll characterize a bit of that and then hand it off to Alli to talk about some of our more recent efforts as well. Just one more comment on our approach to supporting infectious disease research and response. We're really committed to a bottoms-up approach. Our engagements tend to be with local or state health departments as opposed to global or national entities as a starting point, really wanting to enable and deliver that capability and capacity at a local level, and then see that those impacts roll up to a higher level as well. Just a little bit of background on the work that we were doing from this about May of 2020 to we discontinued this effort in June of 2021, and our support of health departments throughout California in what we called our COVID tracker project. This is what I was thinking of as an end-to-end program for genomic epidemiology, essentially as a service. Health departments could send their samples directly to Biohub. We would sequence them. We would put them through our bioinformatics pipelines, conduct QC, assemble genomes. And then really we're committed to ensuring that those data could be used effectively in the response as well. So we built phylogenetic trees, and then critically had staff committed to sitting down with the laboratorians, with the epidemiologists and health officers to walk them through the data that they were receiving, being sure that they understood it and were comfortable with it, appreciating that for most of these groups, particularly at the local level, these were completely new data streams that they hadn't worked with before, and wanted them to be comfortable with that interpretation to where they could turn around and take action off of that. One of the side effects of that work that was also of a lot of value to us in being able to identify our priorities moving forward and how we could contribute in the future was being party to the learning process for these local users as well. It gave us a direct view into what their needs are, where there are gaps in their systems currently, and where we might be able to help enable those capabilities moving forward. As we've rolled into 2021, we've really refocused our efforts as opposed to being the primary service provider into being an enabler for these health departments to be able to stand up these capabilities on their own. So we've done that through a mix of training and technology development, which we'll talk a little bit more as we move forward as well. So the outputs of the COVID Tracker project, we ended up working with 22 health departments in California. The US public health system is very distributed and federated. So each of these jurisdictions holds local public health authority. So you're working with each of them in a bilateral capacity, as well as at the state level. And a lot of the barriers that they had at the time, there's about 15 public health laboratories throughout California. Just a handful of them had the capacity to conduct whole genome sequencing at the beginning of the pandemic. So one, we wanted to fill that gap and enable them to have those data available. We generated a bit over 13,000 consensus sequences while we were operating as a primary service provider. That accounted for a pretty large proportion of what was being conducted in the United States early in the pandemic as well. And then had hundreds of hours of hands-on training and interpretation to help our public health partners be able to actually interpret and apply those data that we were returning to them as well. What we saw probably is not too surprising to anyone on this call. A lot of the bread and butter genomic epidemiology in an applied setting on the front lines, but allowing the DPHs to determine in a very granular way where transmission is taking place within their communities, how they can target interventions to disrupt those transmission chains, and then also evaluate the efficacy of those interventions once they're applied. As we roll into 2021 as well, a lot more emphasis on thinking about variants and potential variants of concern and how those are impacting their communities. So as I said, as we stepped into 2021, really wanted to refocus our efforts on, okay, nobody thinks it makes sense for us at the Chan Zuckerberg Institutes to be the ones conducting genomic epidemiology in the long term, but we're committed to helping our partners stand up this capability, and really saw an opportunity in two places. One is capacity building, which I'll hand over to Alli to talk about now. And the other, I'll just mention, which we'll speak to later in the presentation as well, is technology development and developing some software tools to fill some of those gaps that we identified as well. One of the unique features about the Chan Zuckerberg Initiative as a philanthropic endeavor is that we carry the hypothesis that there is a role for technology development in the philanthropy space. So we have a full suite of product development and engineering software teams that are committed to developing software, in this case in the scientific space, to support researchers and responders on the front lines as well. So with that, Alli, I'll hand it over to you, and just let me know when you want me to click through slides.
Sounds good. Thank you, Patrick. Yeah, so as Patrick was saying, the COVID Tracker Project was this incredible opportunity to help support public health action and genomic epidemiology at the local level. And I think where this is really special is, as many of us have seen over the pandemic, sequence data have been incredibly important in providing surveillance information and situational awareness. But one of the great things about deploying genomic surveillance and genomic epidemiology capacity to frontline agencies is that it can really guide their policies and inform their public health practice in a way that enables them to intervene and interrupt transmission. And that's a pretty special thing. And so I just wanted to walk through a couple of concrete examples of how different counties that we've partnered with have used this information. So in this first case, this was a question regarding the effectiveness of a screening program that was implemented at a county jail. So, often, a jail is a congregate setting whereby SARS-CoV-2 can transmit quite readily if it is introduced. And so in this particular county, the local public health officials were wanting to ensure that they prevented the introduction of new cases of SARS-CoV-2 into the standing residential unit of this particular jail. So they had a screening and quarantine program in place whereby newly incarcerated individuals were tested if they consented to it, and also placed into a 2-week quarantine before being put into the standard residential pod system of the jail. And this screening program was in place, but the jail had also had a large outbreak, and they were continuing to see incident cases after they had thought that the outbreak was wrapped up. And this resulted in this question of, okay, well, we're seeing this continuing trickling of cases. Is that because we actually haven't fully stopped this outbreak that we believe we brought under control? Or are we in fact reseeding the jail with new outbreaks? And is our screening program that we worked so hard to design not actually effective in preventing these introduction events, which we're trying to prevent? Next slide, please, Patrick. Thanks. So this is sequencing data on a phylogeny. The samples in red are sequences that were collected from individuals who tested positive during their quarantine period as part of the intake screening program. And you can see that they are distributed throughout the phylogeny. They are related to the diversity that is circulating in the background community. And you can also see that they are terminal tips. So there are not additional branches that are descending from those infections, which is a really good indicator that these are sequences or these are infections that are related to transmission that's occurring in the community. And it's not actually seeding ongoing transmission chains. In contrast, when sequences were derived from individuals who tested positive in the residential pods of the jail, those are all colored in pink, and you can see a large cluster at the bottom of this tree in which many of the samples are identical, in which there's a bit of accrued diversity as well. The other thing that you'll note is that these samples cluster in a different portion of the tree. They're not actually related to any of the intake samples genomically. And this provides really good indication that the outbreak that was occurring in the residential pod and these incident cases that were being detected was a result of this particular outbreak not actually being fully controlled within the jail system and not the result of reseeding events. And so this showed that this screening program actually was effective in preventing introductions of SARS-CoV-2 into the jail among the incarcerated population, but that it wasn't preventing introductions or ongoing outbreak that might have been brought in by other individuals as well. And this allowed the public health authorities to work with the jail to really target their intervention and know that they could be confident in the screening program, but target their efforts more towards limiting the ongoing outbreak that was happening in the jail. So a really tangible way to evaluate your public health systems. Next slide, please, Patrick. So this is a different case from a different county as well. They had an incredibly large outbreak in a skilled nursing facility. And this was a very major outbreak. It happened in the winter of 2020 when we were having a peak in cases in the US and in many places of the world. But this outbreak was notable. It actually had a 100% attack rate within the resident population of this skilled nursing facility, and it affected many of the employees as well. And so the county was very, very interested in trying to understand what happened. They had policies in place to try and prevent outbreaks in congregate settings and very sort of active testing and surveillance policies, but this outbreak still happened, and they wanted to know maybe why, how did this introduction occur? So if you look at the temporal data, the traditional epidemiologic data from this particular case, what you would see is that one of the employees of the skilled nursing facility had tested positive for COVID-19. And about a week later, the additional staff and residents started to begin to test positive as well. And from a traditional epidemiologic standpoint and an epi curve like that really looks like the initial positive employee was the likely index case for this outbreak. But in fact, the genomic data told a little bit of a different picture. Next slide, please. So this is on the right here is a figure showing the genetic diversity of sequences within the community. Those are noted within gray, as well as within the skilled nursing facility. And those are shown with primarily yellow squares, but some red squares as well. And what I want to bring your attention to is that the putative index case, the employee that tested positive about a week before all of the other residents tested positive, their infection was sequenced and it clusters down in the lower portion of the tree. It's actually, this individual has an identical viral genome sequence to another person who was sampled from the community with absolutely no link to the skilled nursing facility. And it clusters within genetic diversity of the virus that was circulating in the community. You can see that here in these additional genotypes. And we've collapsed tips here for sequences that are identical in genotype. So, 6 representatives of that community strain and 2 representatives of the following one. In contrast, if you look at the most basal genotype that was detected 40 times within the SNF, it's actually 4 nucleotides diverged from the sequence of the putative index case, which is far, far more divergence than we would expect to separate cases only separated by 5, 6, 7 days in time. And you can also see that this basal genotype is in fact identical to many community sampled cases without a link to the SNF as well. And there's a fair amount of community transmission and accrual of diversity that's occurring within the community that separates the putative index case from what we see genotype-wise circulating within the skilled nursing facility, which is a really sort of strong indication that these are separate events. And despite the fact that there is this common exposure profile of the SNF as well as very clear temporal signal, in fact, we can use the sequence data to rule out the putative index case. And what that means is we don't actually know how this outbreak started. And while that can be a little bit frustrating, it's also important to, ensure that we don't think we know what happened when we don't actually know what happened. All right, Patrick, next slide, please. So, these were just a couple of examples that I walked through. There's been many, many more of individuals looking at occupational outbreaks, of household transmission analysis, looking at similar exposure histories and understanding whether exposure, the common exposure history is the result of the infection. There's been VOC analysis. There's all sorts of interesting work that's been going on across many of the counties that we've worked with. And I think what's really incredible is it has demonstrated the utility of this genomic surveillance data at the local level in California, where many of these labs were potentially interested in sequence data, but couldn't necessarily get buy-in previously in order to develop their own genomic surveillance. And so really this project helped to give folks a taste of what you could do with these data, and they found it really useful. And what that has meant is that, the partners that we've worked with are very interested now in not just having genomic epidemiology as a service, but really to bring that capacity in-house, which is really where it belongs, right? So genomic data are just another data stream for doing epidemiologic analysis in public health practice, and that should live within public health authorities. Sure, you can go forward, Patrick, thanks. So as probably most people on this call will know, it's great to want a genomic surveillance system. There are a lot of functioning pieces that need to work and work well in order to get that full stack genomic epidemiologic pipeline. So you need to be effectively generating the sequence data within the wet lab. You need to be managing that data and also trying to find ways to link it with the epidemiologic metadata. That can be challenging. As many of us know, especially in phage, there's a lot of bioinformatic work and quality control on the sequence data. Bioinformatics can be very challenging to access, especially if you're not comfortable on the command line or you don't have access to scientific computing infrastructure. And then finally, even if you have all of those pieces put together and you can generate trees with high quality sequence data, and that's happening regularly, you're not going to derive benefit from those analyses unless you have an understanding of how to interpret infectious disease dynamics from phylogenetic trees or other genomic data objects. And so really, all of these pieces need to come together to have a functional genomic epidemiology program within public health practice. Next slide, please. So we're looking at all of these pieces, and as Patrick mentioned, we're tackling this both in a mix of capacity building as well as software development, technology development. So across the full range of these activities within genomic epidemiology, we're providing training and troubleshooting and support. And then we are also working on the data management and bioinformatic and data analysis side to try and build a software ecosystem that enables easier, graphical user-based interface software for doing these analyses that makes this computation more accessible to folks in public health. So I'm going to talk a little bit about the capacity building side of this, and then I'm going to turn it back over to Patrick to talk about software development. Next slide, please. Oops, one more. Thanks. So when it comes to generating the sequence data, the first point in that pipeline, we make resources as well as training readily available to bring folks online with genomic sequencing in their public health labs. So the rapid response team does a lot of protocol development as well as providing all of these guidelines for how to load a sequencing machine and how to interpret QC metrics that Illumina potentially return returns to you. And all of that is made publicly available through their website. So I've taken a screenshot of that there. If you want to Google it, you certainly can and can go there. But all of those laboratory protocols are made readily available. But beyond that, we also provide in-person training. So this is an example from one of the counties that we trained in February of 2020, showing off the coverage plot from one of their newly sequenced genomes. And so we do kind of 2 or 3 day trainings to teach folks how to basically just handle the protocol and generate sequence data themselves. Next slide, please. We also need to follow up on the sequence data, and we support folks in learning how to assess the quality of their data and use some of the bioinformatic tools that are available to them. And typically what we do is that after that wet lab training, we'll walk them through quite slowly how we would do different quality control and bioinformatic analysis on each of the samples. And then we become available for usually a month. It could be as long as necessary, but many of the folks that we've partnered with use this support for about a month. But we'll go through almost every sequencing run that they generate and talk about the quality metrics with them and assess the run. And after about a month, they're usually independent enough in order to feel very comfortable looking at the sequence data, determining whether they're happy with the percent coverage, and also with some of the metrics that Nextgen flags for them about the sequence quality as well. And so that is sort of ongoing training and support as well. Next slide. So we also need to work on this interpretation piece because that can be very challenging. And so to that end, we've made available a lot of didactic materials that teach the fundamentals of genomic epidemiology interpretation. So this program was initially started as a seminar series that was run bi-weekly with the different county level departments of public health that we worked with. But we recorded all of the seminars, and all of those recordings are also publicly available at this URL down here, covidtracker.czbiohub.org/resources. We're currently in the process of reworking this a little bit and making it more into a standard curriculum where the concepts are covered in sort of shorter videos and have clear learning objectives attached to them. But if you are interested in these materials, you can certainly access them right now. And we're hoping to have that new, somewhat revamped introductory course available in the new year. Next slide, please. And the last thing that we do, or maybe not the last thing, but one of the things that we also do is that we try to support inquiry-based learning through regular standing engagements with the county level departments of public health that we partner with. And so what this often looks like is a weekly meeting, or perhaps a meeting every couple of weeks where we discuss any issues that the lab is having that maybe they want to work through troubleshooting wise, or any questions that the surveillance epidemiologists have. And we work through trees and we talk about them and how one could interpret them and what additional analyses you might want to do in order to follow up on something more. And this really provides the standing infrastructure for counties to take some of the concepts that they're learning, try to apply to themselves, figure out maybe what's not clear, what additional questions they have, and then be able to come back and have that additional learning and have that circular learning environment in order to support greater independence in analyzing and interpreting these data. And that has been incredibly rewarding, and a lot of the work going into understanding outbreaks and informing policy to interrupt transmission comes out of these types of meetings. And with that, I will hand it back to Patrick to talk about some of the software development work we've been doing as well.
Great, thanks, Alli. So as Alli noted, the area that we're targeting for supporting with the software development piece is really in the bioinformatics and data management piece. And we have an Aspen that we've released to our partners in California since June of this year. And we're just beginning to release that outside of California as well. And really what we're envisioning here is it's a tool for making genomic epidemiology more accessible to people working on the front lines of epidemic surveillance and outbreak response. One of the needs that we certainly saw was, or one of the barriers was people being able to manage these data, the genomic data effectively in these new data streams, and needing to learn a new coding base to do that. So everything that we built within the software platform here does not require knowledge of coding. It's all point and click functionality that we deliver in there. So where does Aspen fit into the ecosystem of genomic epidemiology and generating these genomic data? On this top line here, you can see a lot of what's happening both on the logistics side and upstream of the wet lab, and then within the wet lab itself. So the inputs that Aspen takes currently are consensus genomes, and then that returns to the user lineage and variant calls, as well as options for point and click generation of phylogenetic trees for analysis interpretation. We use both Pangolin and Nextgen for generating the trees. And then obviously, excuse me, the ability to overlay that metadata is very critical for linking the data, linking the genomic data back to actionability. I don't think that's surprising to anyone on this call as well, but just want to highlight that it's really that linkage to metadata that's extremely critical for us to be able to actually make these data actionable for our users. So Aspen does a couple things. It allows people one dashboard where they're able to see all of the samples that have been sequenced from their jurisdictions. One of the challenges that we've seen from a lot of the laboratories is they almost feel like they have genomic data coming from all over the place. They'll be getting reports from half a dozen different laboratories and trying to track and match those. And essentially, we're giving them a single platform to be able to put all of that data in and then easily select which of those that they want to either download for structuring metadata or the FASTA files themselves. And they can also select which samples they want to send to phylogenetic tree builds. And then we have some sensible defaults for people making decisions on the types of trees they want to build and how they want to pull in contextual data to support the inferences for whatever investigation they're performing too. One of the elements that we appreciated as well is that I think I skipped a slide up there too, but just to note that we're building this with public health or Ministry of Health, frontline laboratory and epidemiologist and public health officer in mind. Really want all of those individuals to be able to go into the same system, see the same data, and interact with those analyses that have been developed as well. So as part of that, people have the ability to manage and share the genomic data as well as phylogenetic trees across jurisdictions as needed as well. So just a couple comments on future development and directions. As I said, we've made this available to users throughout California for the last few months. We're beginning to expand that outside of California as well. If you or the groups you're working with are particularly interested in getting the easy generation of phylogenetic trees and management of data, we have contact information at the end of the presentation. Please feel free to reach out. Happy to give you an in-depth demo and see if it would be a good fit for supporting your needs or those of your partners. Really wanting to enable actually making all of these, the huge volume of genomic data that's being generated as actionable as possible and extracting as much value and information out of that as we can. The one other thing that I'll note explicitly is that we do all of this at no cost. We don't charge anyone for either using the platform or for the computational effort that goes into it as well. So a couple comments on where we're anticipating future development later this year and as we roll into 2022 as well. One, we do pull data directly from GISAID to inform the contextual tree builds, as well as data, excuse me, tree builds for the specific jurisdictions as well. So anything that's publicly available in GISAID for California can get pulled into that California build also. We do want to make that integration with the centralized data repositories a bit easier to use and expand that beyond GISAID as well. We're looking into exploratory analysis and visualizations also. You know, I think many of us on this call are probably quite used to looking at phylogenetic trees, but there is a pretty steep learning curve for people stepping in, learning to interpret those de novo. So looking at alternative visualizations that may allow public health users to answer some of their standard questions more easily as well. We do anticipate moving beyond COVID. We currently just support COVID. We expect to move beyond that as well. We're still identifying just which targets we'll start with, but I don't think the primary players rising to the top of the list would surprise any of you either. Certainly want things where we expect the genomic epidemiology would be particularly impactful in the context of public health as well. And then integrating consensus genome generation within Aspen itself, and allowing people to submit to public repositories as well to help ease some of those workflows and burden in there also. So I'll just wrap up there. I want to leave time for questions and discussion, and just to note that we're a very active team who've contributed across both the COVID tracker project as well as Aspen and capacity building, and want to highlight that here. And then finally, and I'll drop these into the chat, but one, if you're curious in learning more about the work, you feel free to reach out to me or Alli directly. You can also reach out to the team at helloaspen@chanzuckerberg.com. And then apologies, we're in the midst of updating our website now, and that'll come out in about a week or two. So this is a long link, but I'll drop it in the chat. But if you are interested in using Aspen, this is just a brief survey to get a sense of where you are, or if you're anticipating a partner group that might be interested in using it, how far along you are in developing your genomic epidemiology related workflows. And we're happy to chat with you about and any of the work we've done here. Alli, anything you want to add there?
No, I think that's great, Patrick. Thanks. I'll drop the URL for the seminar resources that I mentioned in the chat as well for folks who might be interested in that.
Thank you so much. So I saw one question was already asked and answered in the chat. But yeah, I just want to repeat it here, and maybe Patrick can also chime in. Ruth Tim was asking whether there will be a way to submit to other databases from Aspen, or if you see this as more of this is your own in-house collection of data.
Yeah, so currently it operates as a workbench, more of the latter, where people are able to pull in data that either they're generating out of their own laboratories, that's coming in from partner laboratories, or from public repositories. We do expect that, and it probably by next year, we would have the functionality built in for people to submit to those public repositories directly as well.
And I've got a question here from Soyan. What was the compliance process like to host public health data? Was there anything that you had to do there?
Excuse me. Within the US context, it's been, I would say it's been front-loaded effort for us on the development and partner end, where we've largely been operating under public health authorization. So that, we don't own any of these data. We're simply hosting them within a cloud-based platform for our users who retain ownership and access to and authorization for who's able to access those data. So in the COVID tracker project context, for example, where we're working with 22 health departments throughout California, each of those is a bilateral memorandum of understanding where essentially we're operating under their public health authority to generate those data and return them to them. Within the context of Aspen, it's simply a matter of users signing the terms of service as they're executing their account being set up. We have access to the data in the context of supporting their investigations and maintaining the platform, but otherwise users retain ownership of the data and authorization for who's able to access it as well.
Yeah.
And then
Alli, anything
sorry,
you want to
Alli,
add?
go ahead.
Oh, I was just going to add that, like, right now, Aspen, you can't upload identifiable information into Aspen. So we do make it a little bit easier to get, say, the Nextgen formatted metadata overlay file. So if you were to build a tree in Aspen, you can also download a sort of pre-filled template metadata file that has the strain names for all of the leaves in your tree. And then you can add your columns of PII to that, but we don't allow any PII hosting actually within the software itself.
And that does actually feed into a question that I was having because, so let me take this up before we get to Soren's second question, which is I saw that the columns there, they were very much sample-oriented columns. So, I was going to ask whether you were adopting a metadata scheme like the one developed by PHA4GE, but I guess because that has quite considerable scope for PII and things like that, that's outside of the scope of what you're doing with Aspen at the moment, right?
Yeah, so we definitely, so I should say I'm on the PHA4GE data structures working group, which means that we certainly have pretty heavily promoted the PHA4GE metadata schema and trying, to encourage folks to organize their data in-house around that such that it is easier, more future-proof, cleaner, kind of all of the good things that you get out of a metadata schema. Since that data does not actually sit within Aspen, we don't necessarily adhere to, we don't support it, but in terms of the capacity building efforts, we're really trying to encourage thinking about data storage and data standardization according to those metrics.
So I guess we can get on to Soren's second question, which is Aspen open source?
Yes, so everything we develop in Aspen is open source and have everything available in GitHub if people are interested in looking into more details there as well.
And you said that it's cloud hosted. Which cloud in particular are you using?
Yeah, we're based out of AWS. We do have partners that use other cloud services as well, and we're able to work with them. And it, we don't need partner groups to get onto that same platform also.
And then if I looked at the trees, it looked like you were using Nextgen. It looked very Nextgen-like. And then, yeah. So effectively, do the users then select sequences that are stored in Aspen and choose to then fire up the Nextgen tree building software, or is there something else going on?
Yeah, no. Go ahead, Alli.
Sorry, Patrick. Yeah, that is exactly what's going on. So users can either upload their consensus genomes into Aspen, and that'll be made available for them to build trees with, or actually we have a new feature now whereby they can take an accession number for a sequence in GISAID, and they can also say, I want to include this sequence on my tree without uploading the data to Aspen. And, that works because we are doing this pull down of data from GISAID, which we're using to source contextual data on the trees, which is just, of course, so important for understanding your own outbreak and not drawing wrong inferences if you only look at your own sequences. So essentially you have that dashboard, you have those sequences available to you either in Aspen or by specifying the GISAID ID, and then you can say that you want to build a tree. There's a few different tree types that we support. So you can build an overview tree that'll be more equitably subsampled across your particular jurisdiction so that hopefully you can get a better understanding of strain dynamics through time. Or because we're really interested in supporting these outbreak response type questions, you can build a targeted tree, which pulls in a lot of contextual data around a few sequences of interest that you have selected and pre-specified. And all of that is being handled through Nextgen. It's just how you change some of the parameters of the Nextgen pipeline, like the crowding penalty or the subsampling scheme, but like it's all Nextgen. And then basically we allow users to kick that off. Nextgen pipeline runs, they get a JSON that then they'll see in the Aspen interface. And then if they click on that, they'll get popped out to Nextgen fetches kind of version of Auspice for visualizing the tree. So yes, it is like, we're making it a bit easier to use Nextgen essentially when you don't have access to Unix-based computing or the compute power needed in order to run some of these larger trees.
Thanks. That actually answered some questions I had about, because people always want to build a tree, and I always ask them, well, which kind of tree are you building? And those are two very interesting types. A question here from Supriya Kumar, who asks, have any of the country public health departments written up the experiences? And also, because it will be interesting to hear from their perspective, and also, can you envisage peer-to-peer training where public health departments are actually helping each other solve problems in the future?
Yeah, go ahead, Alli. You've been directly engaged in leading some of those efforts.
So, I mean, we are working, so some of the counties have been very interested in getting their experiences with these data out into the peer-reviewed literature. So currently we have a manuscript with the Humboldt County Department of Public Health up on medRxiv. And that is we just got the reviews back actually last week. So that'll hopefully be coming out. And that was really, Humboldt County really wanted to have a record of how important it was to have sequence data as part of their COVID response. They're notable. They're a pretty rural county. They're low resource. About 20% of their population lives below the poverty line. They're a healthcare provider shortage area. So it's challenging to provide public health response within the county. And the sequence data was incredibly useful to them, and they really wanted that out there. So that's going to come out as a manuscript. We've also worked with some of the county departments of health to write up case studies to really show exactly how they, how a particular outbreak or question was investigated and what the alternative hypotheses were. Something that is not quite as polished as a manuscript, but shows more clearly like what the actual step-by-step train of thought was. In terms of experiences of working as being part of the COVID Tracker project, I don't know that the counties have written up anything kind of expressing what it's been like to be part of that. But hopefully that might come out. I mean, certainly, I think, what we have really seen is that it's, there's certain folks in public health who are very interested in genomic surveillance, and there are other folks who really aren't exactly sure still how these data can be used and how it informs public health decision-making. And I think that demonstrating that and taking that beyond just the academic genomic epi studies that we have and really showing how it guides public health decision-making and interventions and evaluating surveillance systems is something that we're really invested in. So I think that we will continue to be working on that. your point about peer-to-peer training is excellent. We are working to try and make a community of practice so that these different partners can help each other and help support each other. It's been a little bit hard right now because everyone's bandwidth is just so completely maxed out, but I imagine that we will try to move into that more sort of peer-to-peer training mode at some point as well. Patrick, is there anything you'd add to that?
Maybe just say that you know, the gap in the literature that Alli's describing is one that we've appreciated generally as well, where you do see genomic epidemiology described from that academic perspective, but particularly that local frontline public health viewpoint of how they're using it has not proliferated in the same way with it. So we have some engagements with our partners in California where they've published specific outbreaks that they've dealt with and investigations there. And then what's really nice about the work that Alli did with Humboldt County was it really was taking a higher level view of how they're engaging genomic epidemiology surveillance and incorporating that into their response more broadly as well. So I hope we'll see more of those examples coming out down the line from us, from others. I think there's a lot of utility there. And from what we've heard from decision makers and public health departments too, just saying, you know, I don't have anything to take to my leadership and show what the return on investment is for me standing up all these resources. Like they send me a paper that can show me what other groups like us have done with this, not the CDC, not a large, well-resourced state, but a small local jurisdiction.
So one more question in the Q&As. And yeah, this is very interesting. So I saw you said that you can incorporate GISAID data, but Darren Lemmer asked that the GISAID has some fairly strict rules about the use of their data in their repository. Do Aspen users need their own GISAID IDs to be able to include GISAID sequences in the tree?
So right now, there is not a requirement that you have submitted your sequences to GISAID in order to use Aspen. I will say though that everyone that we work with is kind of mandated by state public health to put as much of their sequence data into GISAID, and I think probably increasingly NCBI as soon as possible, and that's just because they are public health agencies within the public health infrastructure of the United States. So that is sort of managed more by the policies of domestic public health. I mean, I think, so yes, GISAID definitely does have some strict rules about how data can be used. And certainly, we're pretty clear that users can't publish on other folks' data. But it's, but yeah, I don't know that we have a way of preventing someone from building a tree that looks at a different, GISAID ID. And really anyone could do that, right? Like I could go into GISAID and I could download a record that was in GISAID and I could, fire up an Nextgen pipeline and run it. It's just that I can't publish on that. And so, those rules are maintained for Aspen users as well. But yeah, I agree. This is a challenging issue. And I think that the sort of GISAID ecosystem and sharing rules are kind of, they're challenging for many parties in this field, definitely.
Yeah, I must admit, I thought that they specifically prohibited combining your own data with their data, but clearly they haven't complained yet, else you wouldn't still have access. Have you added or considered adding support for a tool like Usher to automatically select interesting similar sequences, to the ones that you've put into a tool like Aspen?
Yeah, that's I realized I was remiss in not commenting on that too. We worked closely with the group at UC Santa Cruz, and actually, as of a new release a couple of weeks ago, you can send samples from Aspen directly to Usher. So we have that integration set up. You know, it's a first version of that. I think what you might be describing there as well, Peter, we are interested in thinking about how we can use those very quick Usher outputs to then inform the broader tree build based off the Nextgen pipelines as well. So we're going to be doing some continued development there also.
Yeah, no, that'll be very interesting. I mean, I think that there's so much that we are still learning how to do with this wealth of data. And in fact, this overwhelming wealth of data, between trying to understand transmission across regional boundaries, trying to understand which factors are really driving transmission, you know, trying to understand, you know, the shifting between different categories, like, you know, neighborhoods, socioeconomic groups, workplace groups, and all of these things. So, and it's far too much for most public health agencies to handle on their own. So yeah, this has been very exciting. We are out of time, but thank you so much for your time and seeing what you're busy with, and good luck into the future.
Thank you very much for having us. This has been great.